Hello everyone, this section is for catabolism, chemoorganotrophic flooding processes. After learning this section, you should be able to describe the products of the flooding reactions, and you should list the three types of chemoorganotrophic metabolism. First, the flooding reactions. Organisms use their flooding reactions to convert their carbon, energy, and electron sources into precursor metabolites, ATP, and reducing power respectively. ATP is a primary molecule used to conserve the energy supplied by an energy source. Reducing power are the molecules that serve as a ready supply of electrons for a variety of chemical reactions. And the precursor metabolites. They are small organic molecules that provide the carbon skeletons needed for biosynthesis of important chemical building blocks, such as amino acids. Then let's say catabolism. Almost all the flooding reactions are part of catabolism. Look at this picture. In autotrophs, the precursor metabolites arise from carbon dioxide fission and related pathways. In heterotrophs, the precursor metabolites arise from reactions of the central metabolic. ATP and reducing power are used in anabolic reactions that convert precursor metabolites into monomers and other building blocks. Monomers are used to generate macromolecules, such as proteins, which are assembled into various cell structures. This formula shows the reaction process. The organic substances are degraded to precursor metabolites, ATP, and reducing power. By contrast, these three substances can be synthesized to organic substances again. Then let's say chemoorganotrophic flooding processes. When chemical organotrophs oxidize an organic energy source, the electrons released are accepted by electron carriers, such as NAD and FAD. Then these reduced electron carriers, in turn, donate the electrons to an electron transport chain. The metabolic process is called respiration and may be divided into two different types. The first is aerobic respiration. The final electron acceptor in this process is oxygen. And the other is anaerobic respiration. The terminal acceptor is a different oxidized molecule, such as nitrate, sulfate, carbon dioxide, or ferric ion. And there is also another metabolic process. We call it fermentation. This process uses an electron acceptor that is endogenous and does not involve an electron transport chain. During fermentation, ATP is synthesized almost exclusively by substrate-level phosphorylation. It is a process in which a phosphate is transferred to ADP from a high-energy molecule, such as phosphonol pyruvate. PEP for short, generated by catabolism of the energy source. Organic molecules serve as energy and electron sources for all three fueling processes used by chemoorganotrophs. Look at the picture on the left. It shows the process of respiration and fermentation. In aerobic respiration and anaerobic respiration, the electrons pass through an electron transport chain. This generates a proton motive force, PMF for short, which is used to synthesize most of the cellular ATP by a mechanism called oxidative phosphorylation. A small amount of ATP is made by a process called substrate-level phosphorylation. In aerobic respiration, oxygen is a terminal electron acceptor, whereas in anaerobic respiration, exogenous molecules other than serve as electron acceptors. During fermentation, endogenous organic molecules act as electron acceptors. Electron transport chains do not function, and most organisms synthesize ATP only by substrate-level phosphorylation. Let's see the three steps of biological oxidation. First, produce electrons. Then transfer and accept electrons. Take glucose for example. The glucose is decomposed to pyruvate, carbon dioxide, and water through glycolytic pathways. Electrons are produced. Also, by TCA cycle, EMP pathway, HMP pathway, and ED pathway, electrons can also be given. Here, let's say the EMP pathway. This picture shows EMP pathways. Glucose is phosphorylated at the expense of one ATP, creating glucose 6-phosphate. It is a precursor metabolite and starting molecule for the pentose phosphate pathway. In the following actions, another ATP is consumed to phosphorylate carbon 1 of fructose. Then fructose 1,6-base phosphate is produced. This is the 6-carbon phase. And the following is the 3-carbon phase because fructose 1,6-base phosphate is split into two 3-carbon molecules, one of which is a precursor metabolite. DHAP is readily converted to glyceraldehyde 3-phosphate. Then the glyceraldehyde 3-phosphate is oxidized and simultaneously phosphorylate, creating a high-energy molecule. The electrons released reduce NAD to NADH. So during the following actions, ATP is made by substrate-level phosphorylation. Another precursor metabolite is made. Finally, the oxidative breakdown of one glucose results in the formation of two pyruvate molecules. Pyruvate is one of the most important precursor metabolites. This is a formula for EMP pathway. You can see in this formula, a molecule of glucose is decomposed to two pyruvates and 2 NADH was produced. At the same time, 2 ATP was formed. Let's then say transfer and accept electrons. This picture shows electron transport chains. Electron transport chains, ETC for short, are associated with membranes. Electrons flow from the electron carrier having the most negative reduction potential to the carrier having the most positive reduction potential. During respiratory processes, for example, aerobic respiration, an exogenous molecule such as oxygen serves as the terminal electron acceptor. See the picture on the right. Picture A shows the mitochondrial ETC, and the picture B shows a typical bacterial ETC. Electron transport chains are associated with or are in the plasma membranes and intracytoplasmic membranes of bacterial and archaeal cells. In eukaryotes, they are localized to the internal membranes of mitochondria and chloroplasts. Look at this picture. This picture shows the creation of a protein gradient by the mitochondrial electron transport chain and its use in synthesis of ATP. Electron carriers in the mitochondrial ETC are organized into 4 complexes. As electrons move along the ETC, protons are transported across the membrane, creating a protein gradient. Proton release occurs when electrons are transferred from carriers, such as FMN and coenzyme Q, that carry both electrons and protons to components such as non-hemming ion protons and cytochromes that transport only electrons. Complex 4 pumps protons across the membrane as electrons pass from cytochrome A to oxygen. The number of protons moved across the membrane at each side per pair of electrons transported is still somewhat uncertain. The current consensus is that at least 10 protons must move outward during NADH oxidation. 1 molecule of ATP is synthesized and released from the enzyme ATP synthase for every 3 protons that cross the membrane by passing through it. At last, let's see the differences between aerobic respiration, anaerobic respiration, and fermentation. You can see the differences on electron carriers, electron acceptors, end products, mechanism of energy production, and efficiency of energy production. Okay, that's all for this section. Here is what is for you.